Now you have seen the synthesis of purine. Let's now move towards the degradation of purines. The major part of this degradation takes place in your intestine. And of course, as was the case with the carbohydrate metabolism or the protein metabolism, you saw that the pancreatic juice played very important role. Same with here. The pancreatic acids, the pancreatic fluids, they will take very important role, play very important role in the degradation of these molecules also. They contains the enzymes which break down the nucleotides into the nucleosides and then further into the smaller components. And as for finally, the end product of this metabolism is the uric acid. This is the final product of the catabolism or degradation of the purines. Those are the enzymes which convert nucleotides into smaller components. They break down the nucleotides, and they break down into smaller components the nucleosides. I have told you the major difference between the nucleotide and the nucleoside, that you get rid of the phosphate moieties, and the nucleotide is then the nucleoside. These nucleosides which have been formed now, they are taken up by the cells of the intestine, the enterocytes. Then these nucleosides degrade to give rise to the free bases, and these free bases of course then further degraded to give rise to the final product of their degradation, that is the uric acid. As I've told you that pancreatic enzymes play very important role in the digestion of different molecules, and of course the nucleotides or purines or pyrimidines, they are not an exception here, especially the purines, because we will later tell you that why pyrimidine their degradation is not that big deal. The major component of the degradation of the nucleic acids or nucleotides is the degradation of the purines. So the pancreatic enzyme like the pancreatic ribonucleases and deoxyribonucleases, they both act upon the RNA and DNA respectively, giving rise to the ribonucleosides and deoxyribonucleosides, and they are further then changed from tri to di and di to mononucleosides. And once you get pure purine bases, they are then further converted into the uric acid, and uric acid is then excreted through the kidneys. The formation of uric acid is a very important step for as far as the catabolism of purine is concerned. Accumulation of uric acid in your body is very harmful for the body and give rise to a lots of really clinically severe conditions like gout. And we will talk about these conditions, these clinical conditions later on. So first of all, as far as the formation of the uric acid is concerned, the AMP, adenosine monophosphate, is then, if we go into the detail of this figure, that will be much easier for you to understand. So first of all, adenosine monophosphate is converted into adenosine, and this is then converted into inosine. Same is the case with inosine monophosphate. The inosine then give rise to the formation of hypoxanthine, and this is the adenosine monophosphate. Almost same sort of reaction will also be as far as the degradation of GMP is concerned, that is guanosine monophosphate. So whether AMP or whether the GMP, they at the end they converge to form the xanthine. This xanthine, under the action of the enzyme xanthine oxidase, then give rise to the formation of uric acid, and this uric acid is then released from the body. There are many clinical conditions, diseases which are related with the abnormalities in this metabolism, abnormalities in the excretion of uric acid, but we will talk in detail about these conditions when we will be specifically talking about the clinical problems which arise because of problems in the, or because of the failure in the excretion of uric acid. So with this now you know that how purines are formed and how purines are excreted in the form of uric acid. Now, as far as the formation of uric acid, it's quite a complex pathway, and I need your real focus on it. For the sake of convenience, let's we zoom this figure. Yes, so you can see here this. First of all, you know, two of the major purine molecules, there's the adenosine monophosphate and guanine monophosphates. They underwent different sort of reactions which ultimately leads toward the formation of uric acid. For instance, first of all, AMP is converted into adenosine under the action of nucleases, and then this adenosine is converted into inosine. Same is the case with the adenosine inosine monophosphate also, that is also converted into inosine. Then inosine is converted into hypoxanthine, and keep in mind this is for the AMP. Similar sort of reaction also occurs in the GMP, where GMP is also under the action of nucleotidases. It's changed into guanosine and then guanine. While here the adenosine monophosphate from hypoxanthine, this is changed into the xanthine under the action of xanthine oxidase, while guanine is changed into under the action of guanisinase into xanthine. So both the AMP, both I must say the hypoxanthine and guanine, they converge to form the xanthine. This reaction converge to form the xanthine. This xanthine then under the action of xanthine oxidase, another enzyme, very important enzyme, it is converted into the final product of the degradation of purine, the uric acid. As I have told you that uric acid, accumulation of uric acid in your body is always a very harmful thing, and it is related with many ailments, clinical ailments, which we will be discussing in more detail when we will be specifically talking about the failure in the catabolism or metabolism of these purine molecules or even pyrimidine molecules.